Good evening. I would like to start off by thanking ASSI for this opportunity and also wishing our friends from Germany and also all the participating societies a very happy, healthy, and safe 2021. Now, the topic of my talk is on the role of subclinical infection in disc degenerative disease. Now, we all know that disc degeneration is inevitable, but the etiopathogenesis is still a controversy. And the big question mark is on the role of subclinical infection. Now, it all started with this paper in 2001, where Alistair actually demonstrated the presence of bacteria in disc samples from herniation. And this factor got augmented by the paper from Albert, where he showed good results following antibiotic therapy in a certain group of low back pain patients. But this idea is still a raging controversy. And most of the non believers actually strongly believe that the bacteria that is demonstrated in these studies is actually a contamination. But however, there is a strong proof that bacteria can be an initiator and propagator of many diseases. Like, for example, we know H. pylori is the cause for peptic disease. We know that there is a strong association between P. acnes and adhesive capsulitis. And in rheumatoid arthritis, there is a whole amount of organisms which have been proven to initiate and propagate the inflammation. And in many cases of aseptic loosening, which were previously thought to be cement related, are now known to be infection. In 2017, we also presented this paper on the Easels Prize winning paper on the role of possibility of subclinical infection being a cause for disc disease. We not only demonstrated bacterial proteins in normal discs, but to a high level in discs with disc herniations and disc degeneration. But there is still a lot of controversy, and this controversy is between infection and contamination. Now, if we have to settle this, there are many missing links that have to be addressed. And I would like to present our work on this subject. First of all, we need to document the presence of bacteria in the discs. We need to decide whether it is contamination or actually infection by showing viability of these bacteria and that there is an infective process going on, and this is the cause for disc disease. So it all starts with the primary question: does normal disc have bacteria? Now, most of us strongly believe that the discs are highly sterile in health because they are the largest avascular structures in our body. But our understanding of infection etiology has always depended on our ability to identify organisms. So far as we restrict to culture sensitive methodology, we will miss identifying a whole lot of microorganisms which cannot be cultured easily. And so metagenomics is and next genome sequencing is the future. And this allows us to identify all the bacteria by DNA sequencing. And this is what we employed. And we looked at 8 MRI normal control discs, 8 degenerative discs, and 8 herniated discs, which were all harvested under sterile precautions and immediately snap frozen. What we found was that there was a rich bacterial presence in all three groups, and even in the normal discs, which showed that even in the health, normal lumbar disc has a good presence of bacteria. But the microbiome of normal discs differed from that of the degenerated and herniated discs, both in biodiversity and abundance. While normal discs had a wide spectrum of bacteria and relatively very low abundance of established pathogens, degenerate discs had a reduction in the spectrum, but they had a relative increase in the number of pathogens. Beneficial bacteria like Bacillus coagulans and Bacillus clausii, they were all present only in organ donor discs. And Lactobacillus mucosae and Sphingomonas were present in abundance only in the organ donor discs. Whereas bacteria with pathogenic potential, like Pseudomonas fragile, Prevetola, and Streptococcus and Prevetola pallens, were all present only in the degenerated and herniated discs. And this is what we reported last year in the European Spine Journal, that human intervertebral discs harbor a unique microbiome, and dysbiosis determines health and disease. Now, what is meant by dysbiosis? Dysbiosis is the balance in the normal between the type and quantity of bacteria which is present in a tissue, and an alteration of the normal ratio can actually determine and cause a disease. So we have symbiotic bacteria, commensals, and pathogens. And when they are in balance, it is the state of health. And an imbalance between the type of organism present can actually cause a dysbiosis, which can result in a problem. Now, in the last 2019 NAS Outstanding Paper Award, we established that there is an inflammaging, a constant low level of inflammation occurring in diseased discs. And we also showed that this could be because of a bacterial induced inflammation. Now, if you looked at normal discs in healthy young discs, most of the proteins that were present were structural proteins and metabolic proteins, and there was no complement cascade proteins or degradative proteins. Absence of inflammatory markers and degeneration was in the young and normal discs. Contrast to this, in degenerated discs, we found an upregulation of complements and degradation proteins and inadequate complement inhibitors, which actually showed that this inflammation which is present in the degeneration and herniation could be as a result of bacteria. In the third part of our study, there was further proof because we established the presence of host defense response proteins. Now we know that whenever there is a pathogen exposed to the tissue, the human body mounts up a defense. And the proof of this can be found from the presence of human defense response proteins. Now, you could see here, we were able to establish a whole number of host defense response proteins. For example, we found that prolactin-induced protein, it was 9 times higher than what was present in the normal disc. And we also know that these act as the first line of defense against invading pathogens. Similarly, complement component and heat tolerant, there was a 3.6-fold variation. And we know that these are all a part of membrane attack complex on bacterial cell membranes, in which beta subunit helps in complement mediated bacterial clean. Proof of all presence of these bacteria actually stand as a proof for infective pathway and activity going on. For example, defensin was also uniquely present only in disc degeneration and in herniation. And defensin is a family of antimicrobial and cytotoxic peptides found in the microbicidal granules of neutrophils that helps in phagocyte-mediated host defense. And the presence of this and another 42 host defense response proteins in diseased discs actually showed proof of inflammation, and an inflammation that is initiated and progressive because probably of bacteria and infection. So we came up with a unified pathway. Most of us used to think that the traditional hypothesis that degeneration is mechanically induced, but we do think that this is an important factor, and this can actually be the initiator of a cascade of events. Mechanical disruption can either lead to end plate breaks or annular tears, both of which lead to neovascularization of the disc. There is ample proof for this in literature. Neovascularization then carries the bacterial, and this can lead to bacterial inoculation, and that can lead to a low-grade infection. And this low-grade infection can lead to a constant source of inflammation. And this inflammatory cascade and perpetuation of inflammation can lead to disc degeneration and herniation. Now, there can be a genetic susceptibility for this, and that is the way that genetics also will play an important part in disc degeneration. So, our study over a period of time has shown proof that bacteria is present in even health, and they are present in a difference of ratio with dysbiosis in the degenerated disc. It is not a contamination because we have proved that the virulence, that they are multiplying, they are viable, and actually they are a cause for infection because we have also proved that there is dysbiosis and there is inflammation and the presence of a host defense response proteins, which actually shows an infective process. We know that human cells versus bacteria in the body, we are outnumbered by the bacteria by a ratio of 10 is to 1. Many of the bacteria which we thought were commensals, now we know that they can be a source of many kinds of disease. Now, it was Chanakya who actually said to underestimate the enemy and to mistake an enemy as a friend are the two mistakes that any king must avoid. So I think in the recent few years, we have now come to the state, mainly also because of the fact and the ability of us to identify all the bacteria that are present in the tissues by next genome sequencing, that many of these bacteria are the source of a constant inflammation in the tissues. And this is also probably the cause, or at least one of the causes for disc degeneration. Now, this is will be very important because if the role of bacteria as it is being established in health and disease, and if we can prove that there is a role for infection in disc degeneration, it will dramatically change the way we look at this pathology, and it will also offer and open up new possibilities for treatment of these. As Louis Pasteur said, we are all on the verge of mysteries, and the veil is getting thinner and thinner. And hope that the truth of what is the real pathology behind disc degeneration will be in our hand very soon. So, thank you very much again for this opportunity, and my greetings to all of you.
I think, thank you, Raja. I think we'll have some questions, if any, from the our panelists as well as from the delegates who are attending the meeting. Before that, may I ask Raja, as you rightly said, there are believers and non-believers, and only time will tell what you're saying. I'm not very
Non-believer.
inclined to, sorry?
You're a non-believer.
At the moment I'm a non-believer. I have, because I understand that yes, in the cervical spine and in the lumbar spine there is more degeneration. So yes, there is a chance that because of the mechanical problems that you have mentioned, the bacteria can go there and multiply. How about an isolated thoracic disc? Right, we see a young disc coming out, right? Sorry, a virgin disc coming out and then causing pressure. So there is not much mechanical instability there, there's not much tear there, but then still it comes out. And the same thing happens in, sometimes we see in children, I mean children in the age group between 13 to 15 or 16, sometimes coming to us with big discs, though rare, they come. And we don't see much degeneration of the end plates there, but we see a nice, soft disc coming out. So does your theory apply in those situations as well?
Yeah, Rahul. First of all, thank you very much for this question. Now, if we say that subclinical infection is the only cause for disc degeneration, that would be very, very wrong. Now, certainly there is a huge mechanical element to disc degeneration, but also mechanical effect also cannot explain everything. For example, you see a degeneration at L4-5 and L2-3, and you get a L3-4 disc, which is absolutely normal. You get various patterns of degeneration with completely normal discs and discs which are completely degenerated. Which gene would affect only one disc in the body, or what is the mechanical effect that would affect L23 but not L34? So it's a very big interrelated issue. And as I told you in one of the slides, so now we think that it is all very, very interrelated. Now, we are now able to identify many of these bacteria also as coming from the gut. So, what we feel is that there are small end plate breaks and also annular ruptures, and that is mechanically originated. Now, once this happens, neovascularization takes place, which takes the bacteria inside. And these bacteria are not staphylococcus or pseudomonas or klebsiella. They are fastidious organisms which cannot be cultured. They can be only identified by next genome sequencing. And they actually are very, very slow growing organisms, and they produce enzymes which can disrupt the collagen, make the disc slightly weaker, which again makes it more susceptible for mechanical effects. So it is not just subclinical infection, it's a combination of what's happening between the mechanics, and also we are saying that subclinical infection can be one of the causes. What we would like to emphasize from our research is move away from the much believed thought process that in health the human discs are completely sterile. It is not. It is just full of bacteria, but those bacteria are completely different from the ones in which we see in the disc degeneration. This is called dysbiosis. And dysbiosis has been proved to be a problem in atherosclerosis, in irritable bowel syndrome, and subclinical infection has been proved in many, many diseases like rheumatoid arthritis or even Parkinsonism now, and many of the neurodegenerative disorders. So, slowly, one by one, many of these diseases are going to be proved at least to have an effect of bacterial infection. So, we are not saying bacterial infection is the end of it all or the beginning of it all, but there is an element in which the subclinical infection produces inflammaging, and this inflammaging weakens the matrix of the disc.
Now, if I can take it further, this question now, how much the number of discs that you operate or you see in the clinical practice, how many of these disc degenerations are bulges or sequestrations are due to infection? Just to put a a percentage, and how many are due to degeneration and other genetic issues in your
All of them are degenerated, and
right?
all of them have bacteria. None of them will grow a bacteria if you culture,
Right.
because the organisms you are talking about are not culturable. So if you tell me, okay, Raja, I sent 10 of my herniated samples and none of them grew any bacteria, I'm sure you're right because they are not culturable. That is the most important problem. These bacteria are completely different from the bacteria which we know as pathogens, and they can be only identified by next genome sequencing. For example, Raghav, we have 1 trillion human cells, but we have 13 trillion bacteria in the body. 65% of these bacteria have still not been coded. So our genome is overridden by the bacterial genome, and many, many diseases are now being associated with this. So let me make it very clear. I'm not saying that bacterial infection is the only cause. I'm just saying that we need to keep sure, have the knowledge that human discs are loaded with bacteria in health and also in disease, and there is a dysbiosis between them. And we have been able to prove by human defense reaction proteins that there is a mild element of infective process and a host response going on. So we are not just saying that is it. We are saying let's think of this possibility also.
Raja, there is a question from a delegate. Similar to the pathogenesis of gastric ulcers and H. pylori, did you find a single organism that was implicated and can be targeted with antibiotic therapy? That's a question.
Yeah. So P. agnes is the one that is being highly implicated. P. acnes, I mean, there are loads of papers which have identified P. acnes in the disc, and people are very strong about it. But in our study, we also found P. acnes. But in addition to P. acnes, we have found other bacteria also. Now, that is the next question to find out. Is it just one of these bacteria, or any of these bacteria can just start a small inflammatory trigger? Now, it is not infecting the whole disc. It is just that these bacteria can trigger off an inflammation. And then the inflammation cascade starts. And once this cascade starts, then there are numerous biological pathways which can lead to degeneration. I mean, it's a long story, so I'm just putting it in short. But this is one of the interesting things that is coming up. There is a mounting evidence for this. But as you said, I mean, when you first think about it, it looks impossible and it's very difficult to believe.
Yeah, Dr. Raghav, we need to move forward, but allow me
Right.
to ask one quick question to Dr. Frank. Uh, the certification that you gave, so I believe it is approved by the regulatory authorities in Germany.
In fact, not really. That's currently exactly the thing which we are working on. So the problem is that we do not have a spine surgeon in Germany at the moment. We have either neurosurgeons or ortho and trauma surgeons which do spine surgery, but we do not have an official spine surgeon. And we are currently working on that topic. So do you have an official spine surgeon in India?
Yeah, we have a National Board of Examinations, which runs a fellowship program, 2-year fellowship program. It's not by the National Medical, it's now combined under the National Medical Commission now. But we have around 8 centers which are running fellowship programs. But ASSI also has its own fellowship. So I was only exploring joint fellowship programs or joint certification or something like that. If we can explore on that, maybe we could carry that further.
Would be very interesting. Thank you very much.
Thank you.
Thank you. We will move on to the next speaker. I'm sorry, my pronunciation of the German names is not very good, but Professor Lindquist, if I'm right, on indication, techniques, and limitations of en bloc resection of primary bone tumors of the thoracic spine.
Ladies and